Just to set the stage a little bit, if we look at humans or dogs, or really any animal, like regardless of size, from the biggest to the, no, it's not working. There you go. Smallest, all of these animals have a non-functional selective answer. And they also all have brains, which is very good news if you're a neuroscientist studying brains. And these brains in all of these animals are made up of cells, and depending on which size of animal you are, they're like 100 million cells in mice, or 100 billion cells in the human brain. But if we look at all of these, the cells in all of these animals, the individual cells work in very much the same way in all of these animals. And because of this, we think that by studying, for example, the brains of mice, we can actually learn something that's really important and really valuable for our insight into how the human brain functions, and hopefully to cure, eventually, human neurodegenerative diseases like ALS. In the lab, we use genetic methods which Chris was showing, genetic methods to label individual cells in the brain, and what that allows us to do is to actually put the brains under the microscope and to study these cells individually, to study their structure and to study their function. Here these are drawings that are over 100 years old, and on the left you have some photographs that I took on the microscope somewhat more recently. And a lot of the focus of neuroscience, as the name suggests, and I think the focus of all the other talks so far today is on neurons, which are these cells that are really long. They send